My name's Professor Shaun Collin, and I'm the Director of the Oceans Institute and a Professor within the Oceans Graduate School here at the University of Western Australia. I also lead the Neuroecology Lab here, and we specialise on understanding the neural basis of behaviour and especially different senses with a particular emphasis on vision. Well, as you know, I was fortunate enough and honoured to receive the H. Barry Collin Research Medal, and this paper, a review of the shark visual system, was the byproduct of a lecture that I gave recently. I thought it was very important to write this review on the shark visual system based on a lot of our work recently on how sharks perceive their environment. We've been interested in the evolution of colour vision and dim light vision for some years, and sharks were certainly on our radar because we know very little about how they visualise their environment. I also believe that it's important to understand the evolution of the vertebrate eye and the different adaptations that many animals, including sharks, used to meet their visual demands under different lighting conditions. Like, I hope optometrists will have an open mind and be interested in this work, given that we need to use different animal models to understand how the visual system actually works. By looking at a number and a wide comparative view of different animal models, including sharks, we can understand a lot more about how they have adapted over 400 million years to their visual demands. The human eye is very similar to the shark eye, so we can learn a lot about the human system by looking at the shark model. I also believe that sharks, in addition to bony fishes, lampreys, amphibians and some reptiles, actually have eyes that continually grow throughout life. So this actually provides an advantage over looking at humans, since the eye has the ability to change during development and all through adult life. So we may be able to investigate how changes in the visual system are manifest and potentially even restore vision or deficits in the human system in the fullness of time by looking at some of these more comparative models. Well, there's a lot to do. Despite a lot of research over many years concentrated on some species of sharks, sharks are inherently hard to access. They're large predators, which are necessary for the environment, but quite hard to catch and maintain in captivity. So I believe the next stages are to continue the work, trying to understand the visual processing side of visual behaviour by looking at more species, trying to use behaviour to answer more questions about sensitivity thresholds, etc., how they behave visually under different environments, and also trying to understand some of their deficits. We're particularly interested in the reasons why sharks, in contrast to almost all other vertebrates, do not have the capacity to discriminate colour. So they're considered cone monochromats or colourblind. And maybe by looking further into this part of the visual system in sharks, we may be able to, in fact, try to restore that, and that could be useful for restoring colour vision in humans one day. Well, that was one of our greatest surprises scientifically, that they were colourblind and in fact concentrate on contrast rather than colour to navigate through their environment and target prey. I think another interesting aspect about the shark visual system, and that is a little bit different to the humans, is the presence of a reflective tapetum. This is a reflective device. It's a structure that sits behind the retina and reflects light back through the photoreceptors to increase sensitivity under conditions of dim or very dark conditions. Sharks have very well developed retinal tapetum, and it's in this way that they're able to navigate and become quite active and actively feed in very low light conditions, which I think is one of the reasons that they've actually survived for so many millions of years. Well, look, as neuroscientists, we strive to understand the human condition by looking at animal models. There are various animal models out there, rodents particularly, but more recently zebrafish. But by looking at the examples or the representatives of the earliest jawed vertebrates, the sharks, I believe we can actually uncover even more interesting issues that could relate to human vision. We can understand the constraints that the visual system is under phylogenetically, and we can also try to understand, if at all, how plastic the human system could be with intervention, whether that be pharmaceutical intervention or somehow improving vision in patients that have some sort of deficit. Well, yes, that's correct. I'm a third-generation visual neuroscientist. My grandfather was an optometrist, and my father is not only an optometrist, but also a visual pathologist. I took a slightly different path. My interest initially was in marine biology, but I managed to combine my interests in marine biology with neuroscience and really started to concentrate on the visual system based on great influence from both my father and grandfather, but also a number of my father's colleagues, all of which were visual neuroscientists. So one thing led to another, combined these two different fields, and have enjoyed trying to understand the adaptations of the visual system in different animals from a whole range of very extreme environments pretty well for my whole career. Look, this is a very hard question to ask, actually. Every eye that we look at is trying to probably do a little bit of a different job, depending on if the animal lives in the deep sea or in brightly lit shallow water or even in fresh water. The light conditions are very different underwater than they are in air. So it's very hard to align a perfect comparison between a human eye and any other animal eyes. Most of them use similar principles, but ironically, I think the shark eye is probably one of the best comparators between the human condition, and thereby I think could certainly in the future be a very useful model. Well, actually this took nearly 2 years to put together. Incorporates over 800 species and shows the diversity of distribution patterns in retinal neurons, whether they be ganglion cells or photoreceptors, from a very large range of different vertebrate eyes. And yes, this is still available for people to look at and hopefully progress and ask new questions of how the visual system is adapted for vision in different environments. Well, finally, I'd just like to say what an honour it's been to deliver the H. Barry Collin Research Medal Lecture, to win the medal, obviously, and to bring together much of what is known about the shark visual system, and the opportunity to publish my paper entitled Seen Through the Eyes of an Apex Predator, a comparative analysis of the shark visual system. Thank you.